And again, one of the things one can do is sort of going backward, but Ella's now working with Vitali, looking at the SPR and ESI measurements to look at the effect simultaneously of addition of these peptides on the integrity of the membrane. And again, as I showed before, if you simultaneously add the peptide and take the SPR response, then look at the ESI response, you can track very nicely how the membrane is being perturbed with the electrochemical measurements to the point where you've basically destroyed the membrane. So you can actually track both the concentration of the peptide added as you add it and its response, and then the membrane integrity simultaneously. And because this is a much faster experiment than the neutrons, you can vary pH and other conditions to look for probing this more for a detailed mechanism. Okay, so with that, I sort of gave you a kind of a broad overview of a lot of different projects, but the basic themes of the methods that we're trying to apply. Hoping if part of today's goal was to try to reach out and think about collaborations. I think one of the themes you can see here is with a set of basic tools like assembling these bilayers, developing these custom instruments for the electrochemistry and for the surface plasma resonance, and the unique capabilities we have in our neutron center, that there's lots of opportunities where you can think of projects potentially where there's interest in understanding structure at the surface, understanding membrane responses to proteins as they interact with the surfaces, and understanding different kinds of lipid compositions. I mean, so the sort of the groundwork has been laid for all these kinds of experiments, and we do reach out to a lot of collaborators to get these kind of projects going in terms of the biology. Again, I mentioned people as I went through the talk, but a lot of the examples I showed